The frontier of the 21st century is one that we cannot see, yet it encompasses billions of organisms that we cannot live without. In fact, most of the cells in the human body aren't even human. 90% are microbial. All plants and animals represent only 2 small twigs on a very large tree of life. The rest of the trunk and branches is made up almost entirely of microbes. At the University of Wisconsin-Madison, researchers in the Microbial Sciences building are bioprospecting the largest reservoir of biologic diversity on the planet. They are exploring the mysteries of microbes, seeking basic knowledge that may lead to cures for infectious disease, to safer, more nutritional food, and to new biological methods for producing energy. A revolution is taking place in the largest academic building on the University of Wisconsin-Madison campus. Researchers from multiple disciplines are rubbing shoulders for the first time in the same facility, sparking ideas to redefine the boundaries of the microbial world and to understand and elude dangerous food-borne pathogens, antibiotic-resistant bacteria, and flu pandemics. Scientists are also supplying each other with new intelligence gathered from studying how microbes live symbiotically in balance.
Microbes do so much still we don't understand, and they do it in ways we don't understand. The Microbial Sciences building brings together investigators from the medical school and agriculture into one community, completely integrated to focus on the biology that is shared. We have some very important centers and institutes. The Food Research Institute is the oldest industry consortium on our campus, focused on food safety and food quality. We also have the Great Lakes Bioenergy Research Center. This is a federal research center that is supported by the largest grant award in the history of University of Wisconsin-Madison. This work is focused on unlocking the potential of biomass for our transportation fuel. We don't get to know ahead of time which discovery it will be that will create this revolution, and that's why this investment in basic science is so important.
What we're attempting to do in that building is solve a number of very significant problems that our nation is faced with and our world is faced with. We've seen emergence of a number of very important disease problems. My laboratory, for example, has studied for years the strains of E. coli that contaminate hamburger meat. We're seeking to try to identify ways in which we break the cycle of infection from cattle to humans. In the public press, you hear constantly about the potential emergence of new pandemic strains of flu. Individuals are trying to identify what makes those strains so virulent to begin with and try to develop new therapies or vaccines to prevent the spread of those organisms ever before they become a pandemic.
My lab has studied antibiotic discovery and antibiotic resistance for over 20 years. But what was an intellectual and professional interest became very personal when my mother developed antibiotic-resistant infections and ultimately died of a lung infection caused by antibiotic-resistant bacteria. And the sadness and the irony of watching my mother die of exactly the phenomenon that I study in my lab and being helpless to do anything about it really fuels my research with a passion and a personal caring that I think most people probably don't bring to their research.
The Department of Bacteriology at the University of Wisconsin has historically played an important role in scientific discovery. Bacteriologists here have been looking for new antibiotics since the early 20th century. Today, they play an even more important role in the race to combat antibiotic resistance as the pharmaceutical industry now focuses its research on drugs for chronic conditions.
I wish there were no superbugs, but in fact there's multiple superbugs. The indiscriminate use of antibiotics in our communities has led to increasing amounts of antibiotic resistance. Bacteria can double themselves every 20 minutes, so a mutant is rapidly found that's resistant to an antibiotic.
Why does AstroTurf cause more infections than natural turf? I was an athlete and I was exposed to AstroTurf, and the trainers and the doctors talked about, if you get a scratch or you get a cut or an abrasion, make sure you get it cleaned off right away. And then they briefed us on what could happen, staph infections, from turf burns or whatever. I've read several stories about athletes who sustain a little cut or a little abrasion and, didn't think anything of it. And then, 48 hours later, their life is threatened by this aggressive bacteria. The University of Wisconsin takes staph very seriously. I feel confident to know we have scientists here at the university who are studying the advancement of these antibiotic-resistant bacteria.
The discovery of new antibiotics can come from unexpected sources. In the late 1930s, Professor Ken Raper's lab technician discovered a prolific strain of penicillin on a moldy cantaloupe at a farmer's market. Tomorrow's new antibiotic discoveries will most likely come from the earth as well. The possibilities are staggering. Just 1 teaspoonful of soil may hold billions of bacteria. But it's like looking for a needle in a haystack to culture the right bacteria, the ones that may produce new antibiotics.
The harder question is, how do we find the antibiotics among the bacteria that don't grow on our Petri plates? And that's where we've turned to genetic and genomic methods to isolate the DNA from those organisms. So we never culture them. We circumvent the culturing completely.
The development of new antibiotics has really fallen back onto the academic community. Probably at least half of the people in the building are keeping an eye toward their research as a way of targeting a new antibiotic.
We eat microbes. We drink microbes. Microbes grow in environments like hot springs at very high temperatures, and you can find them in extremely cold places like the Antarctic. Most microbes are good, but only a few cells from a bad microbe can kill you.
My real specific interest and passion in microbiology is focused on neglected diseases of man. The organism that I work with is one that's endemic to sub-Saharan Africa. It's called the African trypanosome. Now, this is a single-cell organism, a protozoan parasite, that's transmitted to man by the bite of the tsetse fly. And it's a master assassin. It kills every host that it infects. I think the practical reasons for being concerned, if not studying these neglected diseases of man, is that it's clear these days that there are no geographical constraints on infectious disease. And we've learned this time and time again. I mean, whether you look at the AIDS virus, whether you look at SARS, whether you look at Ebola, whether you look at malaria, all these different diseases which are or were largely prevalent in other areas of the world are now impacting on this country.
Salmonella outbreaks in the United States food supply make an immediately tangible impact on the nation. Scientists in the UW's Food Research Institute routinely help to fingerprint the source of potential culprits by matching extracted DNA from infected individuals and food sources.
I think there's a greater awareness now of food safety, and particularly as it relates to pathogenic types of E. coli. It's not always easy to specifically identify these pathogens. In a lot of cases, the food product's gone or it's already been eaten. It takes time, which is frustrating for people. If a particular pathogen shows up in multiple states, epidemiologists can then start making associations and linkages between these pathogens and fingerprint types in an outbreak so that we can more rapidly respond to a particular outbreak and recall food and get it out of the market.
Currently in the fast food business, I'm an investor in Burger King and formerly an investor in Taco Bell, and some of these things have affected our business. When you have foodborne problems in any food service business, your business tends to drop off pretty quickly. And obviously you're in the business to provide a service and you're in the business to make a living. And when sales go down, that's a problem. For a scientist who is credible to come out and say, we understand what's happening, we have narrowed down the problem, and this is how we fix this, is important.
I'm very hopeful that 10 years from now, we're going to have a really critical idea of how 1,000 different species of bacteria inhabit our intestine and how they all interact together. It's a very complex problem requiring tremendous computing skills and a lot of creative bench science. So I hope we can understand how beneficial bacteria can help us. In my own lab, I hope I have a really clear understanding why 5% of all women in the world have repeated urinary tract infections. For other investigators in the building, I hope they can provide a way of recognizing why pandemic strains of flu are so deadly, and if these pandemic strains break out again, as history would predict, that we have a way of blocking those pandemics before they ever really catch fire. I hope colleagues will discover new antibiotics, ones that will help us combat MRSA, multiply drug-resistant tuberculosis strains, and the like. I also hope that in 10 years that a number of serious foodborne problems are no longer foodborne problems.
I like to think that before the end of my career, I'll discover the antibiotic that could have saved my mother, and that will be satisfying if it happens because it might help many other people like my mother. So one of the magical things about the Microbial Sciences building is the uniting of very basic researchers who study how cells work and how they're put together and why bacteria do what they do and how they do it, with people who are interested in specific microbiological problems and applications of microbiology to either food, the environment, or an infectious disease. The inside is full of light and openness. It gives me the same kind of feeling that I get when I walk into a truly magnificent church or temple. That sense of reaching towards something greater and bigger. That's what I think of as science, that it's always reaching for something more.